G'day everyone, Jamie Chapman here. I'm going to be giving you a lecture today, an online lecture on the histology of nerves, vessels and lymphatics. This is to sort of get you ready for second semester because you'll begin dissection, and as you do your dissection you'll begin to see a lot of these features, particularly blood vessels, maybe not so much lymphatics. You might see some lymph nodes, and definitely you'll see some nerves, and we'll see that the blood vessels, the arteries, the veins and the nerves often run together in structures known as neurovascular bundles. But this is part one, and in part one we're going to be looking at the histology of the blood vessels. So the learning objective for this part one is that you should be able to compare and contrast the general histological structure and function of the different types of blood vessels found in the body. So of course the circulatory system consists of a series of closed endothelial tubes, and they function to of course distribute oxygenated blood from the heart to the capillary beds in the organs such as the gastrointestinal tract, the limbs and so on, and to return that deoxygenated blood to the heart where it can then be pumped to the lungs to be oxygenated. So in blue here is representing deoxygenated blood, and in red here the oxygenated blood. So we get oxygenation taking place within the alveoli of the lungs, and then that's distributed to the body, bringing oxygen and nutrients to the various parts of our bodies, and then the deoxygenated blood returned to the heart to be taken to the lungs once again. So in this sense we have two types of circulations. We have the systemic circulation, which involves the aorta distributing the blood to the body, and the pulmonary circulation with the pulmonary vein leading to the lungs where oxygenation occurs, bringing it back to the heart. So we've got these two different types of circulations taking place. So blood vessels generally have a very similar arrangement in that they're made up of three different layers, which we actually call tunics. The innermost layer is known as the tunica intima. Tunic means layer. Intima means the intimate layer, so the innermost layer. It's also referred to as tunica interna. So intima, interna are interchangeable terms. The middle layer is called the tunica media, and then the outer layer is known as the tunica adventitia. It's also sometimes referred to as the tunica externa. So the interna is closest to where the blood exists, the lumen next to the blood, and the adventitia is the outer connective tissue layer that binds it to other surrounding structures. And so when we have a look at the circulatory system, leaving the heart, we actually have different types of blood vessels. So we have the elastic arteries, and their name suggests they're very elastic. They're able to expand to accommodate increased volume, and then they naturally recoil, and that helps to maintain your blood pressure and help to move and distribute that blood throughout the body. Then from elastic arteries we move to muscular arteries. These are particularly in the limbs and have a great deal of smooth muscle within their wall, within the tunica media. From there we lead to smaller muscular arteries. Arterioles, this is part of the first part of the microvasculature, so it's usually around about 100 microns or less in diameter, 1 to 2 layers of smooth muscle cells around the outside. Then to the capillaries, where we get the exchanging occurring, oxygen coming in or leaving and supplying oxygen to cells, and nutrients and waste being exchanged as well. Then that blood is then collected by small veins known as venules. Those venules are then drained into small veins, into medium veins, into the large muscular veins such as the vena cava, which drain the blood back to the heart for reoxygenation. So that's the sort of flow on from the heart: elastic arteries, muscular arteries, arterioles, capillaries, venules, veins, and then back to the heart. Now if we have a look at the comparison here, we've got the aorta, which is an elastic artery. We've got muscular arteries, arterioles here, capillaries. We've got their sizes, general sizes here, venules, veins, and the vena cava. You can see the vena cava is quite a large vessel, as is the aorta. If we have a look at the blood pressure within these vessels, you can see within the elastic arteries very high blood pressure 120 on 80. So here's our systolic level when the heart's in full contraction. Diastolic level is when the heart's in relaxation. So quite high levels until you reach about the level of the arterioles and it begins to drop. Then we've got lower pressure here down to about 20 millimeters of mercury here in the capillaries, and then that continues to fall through the veins, and very low pressure, blood pressure within the large veins, the vena cava and so on. So somehow these veins need to still return the blood to the heart despite the fact that they're under very low pressure. So there's less sort of pushing that blood to go back to the heart. So these vessels actually have various structural features to help them return the blood to the heart under this low pressure. So as I mentioned, this is the general histological arrangement of blood vessels, and we've got in comparison the artery and the vein side by side. So the tunica intima, or the tunica interna, consists of a simple squamous endothelium. Generally, we've got a little bit of loose connective tissue there as well. Of course, every epithelium has an underlying layer of loose connective tissue. The tunica media, this is this layer here, represented here. In the arteries, it's the largest of the three layers and consists either of smooth muscle and or elastic tissue, and depending on the blood vessel that it's associated with. And then the tunica adventitia, or the tunica externa, consists of connective tissue, and depending on the size of the vessel, you may find small blood vessels out there as well, and we'll talk about why that is in a little while. These are known as vaso vasorum, which means vessel of the vessel. Now, just be aware, all arteries, arterioles, venules and veins have these three layers. Capillaries don't. Capillaries actually are just little layers of tunica intima with an endothelium and a little layer of loose connective tissue beneath them. So these are sort of my take-home features, my tips about identifying arteries and veins in histological sections. So these are sort of not taken out of the textbooks. This is my personal interpretation of things to look for when you're looking at a histological section and you're trying to determine whether you're looking at an artery or a vein. So in general, arteries have thicker walls and are more rounded in shape than veins. So veins are classically irregular in shape and have a much thinner wall than their accompanying arteries. Arteries then have much thicker walls, usually with more smooth muscle, particularly if we're talking more peripherally, and are more rounded in shape than veins. So arteries conduct the blood away from the heart. They're more round in shape and have proportionately more smooth muscle and elastic tissue than veins of comparable sizes. The tunica media of an artery is its largest and most functionally significant layer. So the elastic lamina that may exist there, or the smooth muscle that exists within that tunica media layer, are helping that blood vessel to perform its function, either in expanding and then naturally recoiling to help maintain blood pressure in the case of an elastic artery, or constricting to regulate blood flow to an organ in the case of a muscular artery. Veins return blood to the heart, and they have thinner walls and are more irregular in shape than arteries. So this is sort of the opposite of what we just mentioned about the arteries. That's a feature that you can use to help identify whether that's an artery or a vein. Veins may have valves. Now these are extensions of the tunica intima, and they help to break the blood up into smaller pieces, so it's much more manageable to move a smaller block of blood than a whole long tube of blood, for example, and that's why they have valves. It sort of prevents backflow of blood and also helps to block the blood up into smaller pieces that's much more manageable to move than a big long column of blood. It's usually much more difficult to distinguish the different layers of tunics in veins. So identifying where the tunica intima ends and the tunica media starts can be very difficult. Whereas in muscular arteries, for example, we've got a nice delineating tunica internal elastic lamina, which is a little wavy line, which we'll point out in a little while. That really helps to delineate between the tunica intima and the tunica media, and I'll point what that means in a little while. Now, as most of your blood at any point in time exists within your veins, blood cells are more likely to be found in veins than arteries in histological section. Now, that's not to say you won't find arteries with blood cells in them, but the blood vessel that's more likely to have blood cells inside it is the veins, because the blood's there for longer. It tends to pool a bit longer in the veins. So we're going to have a quick look at each of the different types of blood vessels and their major features, and then that will end this part of the lecture here. So the biggest and the first of the arteries that leave the heart are the elastic arteries. So these are the largest ones, including the aorta, the common carotid, subclavian, brachiocephalic arteries. So they, of course, with the contraction of the heart, receive the blood under high pressure from the heart, and then through their natural recoil, this helps to conduct that blood through the rest of the body. So they're characterized by having a very thick tunica media. So here we've got a trichrome stain section. Blue is collagen. Red here are the elastic fibers or elastic lamina. And you've got a little epithelial lining on the surface here of the tunica intima. So very small tunica intima, very massive tunica media, and then an adventitia on the outside here. And this tunica media, because of its name, there's lots and lots of these bundles of elastic fibers called elastic lamella or elastic lamina. Doesn't really matter which terminology you use, but they're thick bundles of elastic fibers, like having dozens and dozens of elastic bands around in the wall of this structure, so that when this expands to accommodate the blood, those elastic bands recoil naturally and help to maintain diastolic pressure. So this passive recoil helps to maintain the blood pressure during ventricular diastole, which of course is when the heart relaxes and begins to refill. So we've got the common sort of blood pressure is 120 on 80 millimeters of mercury. 120 is the systolic pressure, so when the heart's at full contraction, and the relaxation, the diastolic pressure, is 80 millimeters of mercury. This 80 millimeters of mercury is brought about by this natural recoil of all of these elastic lamina in these elastic arteries. Now, one thing you might notice in this blood vessel, certainly out in the tunica adventitia out here, is a small blood vessel, so a couple of blood vessels here, and then inside here as well we've actually got some blood vessels. These are the vasa vasorum. And you think about it, this is a thick wall structure. Diffusion's only going to work so far to allow nutrients to be provided to the smooth muscle cells that are found within the tunica media here. And so in order to provide nutrients and get rid of waste for the outer layers of the wall, we have little blood vessels of this blood vessel, and they provide nutrients and help to eliminate waste in this outer layer of these larger vessels. And the large veins also may have vasa vasorum. So that's these little blood vessels of the blood vessels. Muscular arteries, examples are in the limbs, so the radial femoral arteries. These are distributing vessels. They have predominantly smooth muscle within their tunica media. So we have these circularly arranged layers of smooth muscle. One thing you might notice about muscular arteries, they often have this very wavy appearance to their tunica intima. And that's because there's a band of elastic lamina present here, known as the internal elastic lamella or the internal elastic lamina, and that creates this waviness related to the tunica intima. Very classic feature of muscular arteries. So if you ever see this waviness, then it must be a muscular artery. That's a really defining feature of them. So this wavy tunica intima, there's an elastic bundle between the tunica intima and the tunica media. It's part of the tunica intima. It's called the internal elastic lamina. It's this wavy line here. So the tunica intima of these vessels consists of an endothelium, a small layer of loose connective tissue, and the internal elastic lamina. Then you have the tunica media, which is several layers of circularly arranged smooth muscle, and then you've got the tunica adventitia on the outside. You can see there's no vasa vasorum here because these are small enough blood vessels that diffusion works from the lumen there. So the arterioles are considered part of the microvasculature. So they're about 100 or 200 microns or so. They help to regulate blood flow through the capillary bed, and they're important regulators of systemic blood pressure. So we've got here we've got the endothelial cells, so of the tunica intima. We've got a little bit of loose connective tissue, and then we've got the tunica media here, consisting of 1 or 2 layers of smooth muscle, and then we've got the tunica adventitia on the outside. This is a nice longitudinal section. So here we've got the simple squamous endothelium. Here's our endothelial cells. There'd be a basement membrane there, a little bit of loose connective tissue, and then we've got 1 layer of smooth muscle cells. And these are running around the outside in a circular arrangement, but when we cut this blood vessel longitudinally, they appear in cross sections. So we know that not every smooth muscle cell is going to have a nucleus in its center in the plane of section, but you can see them here. So here's a arteriole under the electron microscope. So here's the lumen over on the right hand side. Here we've got our endothelial cells. You might see a few endocytotic vesicles here. There'd be a basal lamina there, a little bit of loose connective tissue for the tunica intima. Then the tunica media here consists of 2 layers of smooth muscles. So here's our smooth muscle cell. You can see some of these dense bodies on the outside. Out here, a few caveolae around the outside here. So here's our tunica media, and then our tunica adventitia. Interestingly, I'm not entirely sure why this happens, but there's quite a intimate relationship between the smooth muscle cells and the endothelial cells. You see they have these little cytoplasmic extensions which connects them. I'm not sure exactly why that is, but I thought it was a fascinating little feature there. So from the capillary cell, from the arterioles, we then move to the capillaries, which are, of course, our exchange vessels. And they're the type of vessel that doesn't have the 3 layers. They only have the tunica intima layer, which consists of an endothelial cell layer and a layer of loose connective tissue beneath it. They're about 5 to 10 microns on diameter. So if you remember, a red blood cell is about 7.5. So you can see it's just fitting here. Sometimes the smaller blood vessels, the red blood cells actually need to squeeze through the capillaries, so they can be a little bit smaller. And the flexible nature of the red blood cells is important for allowing to flow through these capillaries. Now, a number of capillaries may have little stem cells associated with them called pericytes. These are undifferentiated stem cells that can become differentiated and eventually form new endothelial cells or new smooth muscle cells if you're going to have a little outgrowth of an arteriole, for example. They're little contractile cells as well, and they can actually contract and regulate blood flow within some of these capillaries. So here we've got a transmission electron micrograph of our endothelial cell capillary, red blood cell filling the entire lumen here. You can see lots of endocytotic vesicles, these little white vesicles here. And then on the outside we've got this cell, and this is actually a pericyte sitting on the outside of this capillary. And if you sort of follow along the basal lamina there, you can actually see it's actually contained within the basal lamina of this capillary. So from the capillaries we go to venules and to veins. You can see what I was talking about it having a much thinner wall, much more irregular in shape, not a nice round shape, and we've got lots of blood cells inside it. Generally a large lumen, irregular in shape, and this relatively thin, very difficult to distinguish the different layers of the wall there. The primary function, of course, is to return deoxygenated blood to the heart. This function is aided by smooth muscle and the tunica media, so you can see some of that smooth muscle here. Valves, which I'll show you some pictures of in a little while. There's no valves present in this picture. Contraction of surrounding skeletal muscle. So the veins are often in the limbs. And when you walk around, your skeletal muscles of your limbs contract, and that helps to squeeze that blood in those veins and help to return it to the heart. And that's helped by the valves, which help to break it up into smaller blocks of blood, but also prevents backflow of blood as well. It has few smooth muscle cells in the tunica media, and the tunica adventitia is generally the thickest of the layers that's binding it to surrounding connective tissue. So you'll note the relatively large lumen, the irregular shape of this blood vessel, only a few layers of smooth muscle despite it being a relatively large vessel, blood cells within the lumen, and there's no internal elastic lamina. So you can see smooth muscle in the tunica media, but you remember there's no waviness associated with the internal elastic lamina, and there's none of that there, so this is classically a vein. So here's a couple more pictures of these veins that we found. So out here we've actually got an artery. So this is a muscular artery. Look at the waviness there, and this is the Orsian stain, which stains up elastic fibers. So the internal elastic lamina there quite clearly defined, and this is all smooth muscle. And then over here we've got the vein, very irregular in shape, not very thick wall, but then we've got these weird little flappy things inside the lumen here, and these are the valves. So they're extensions of the tunica intima, and they help to block the blood up and prevent backflow. And this is a really nice micrograph here. I've got a bigger version of it. So here's one of our valves. These are extensions of the tunica intima, as I mentioned, and it's really showing its role here. So here it's actually blocking the blood up, preventing backflow, and then here we've got the clearer lumen. You can see some of the cells starting to leak through there. So the large muscular veins include the vena cava. Now these are very unique blood vessels. Rather than having lots of smooth muscle in the tunica media, the large muscular veins like the vena cava actually have most of their smooth muscle in the tunica adventitia. And as I say, this is very bizarre and very unique to these vena cava. And so this smooth muscle here, this is actually arranged longitudinally. Now remember, all of the other smooth muscle in the other blood vessels is arranged circularly, but here in the large muscular veins, they're actually arranged longitudinally, and that helps to cause this peristaltic pumping of blood. Remembering at this point, there's very, very low blood pressure in these blood vessels. And by this stage, the vena cava, we've moved out of the limbs, so they can't get that help from the skeletal muscle contraction to return the blood. So what it does is this longitudinal layer of smooth muscle contracts and forms this peristaltic movement of blood, helping to oppose the action of gravity. So as I say, this arrangement, structural arrangement in this blood vessel of having smooth muscle predominantly within the tunica adventitia and longitudinally arranged is specific to the vena cava. So as we know, often the arteries travel together with veins, and the arteries and veins often travel together with nerves. So here we've got this small section here. We've got the artery. You can see nice round lumen. Here we've got the vein, and then we've got a couple of little valves here present, and then over here we've got the nerves. So we often form these structures known as neurovascular bundles. They often run in threes, although they often actually run in fours, with the lymphatic vessels often coinciding at the same time. We'll talk about what lymphatic vessels are in part 2. So this occurs at the broad level, so the big level, like the femoral artery, femoral vein, femoral nerve, but it also occurs at the level of the arteriole venules. So these are actually more likely to be arterioles and venules rather than arteries and veins, which is why you don't have that waviness which we typically see in a muscular artery. And here's our peripheral nerve and a little lymphatic vessel there. Alright, so that's it for part 1. That's our histology overview of the blood vessels. Now, please, before you move to part 2, take a chance and have a go at the formative part 1 quiz, and this will provide you feedback on your level of understanding of this part. And of course, feel free to attempt these quizzes as many times as you like for your own revision purposes. I'll see you in part 2.